presentation. So it's really my pleasure to be here tonight, actually, this evening with you, to share with you some of our new work on the analysis of fragment library. Okay, I've lost something. Okay, it seems
I just turned off my cam.
Okay, that's why something weird happened. But do you still see everything correctly?
Yes.
Yes. No answer, so I go on. So before I start, I just would like to mention that what I will present today is mainly the work of Julia Reviro in Vernon, who is PhD student in my group, and her project is about the design of new antibiotics. And this work was published in December last year in RC Medicinal Chemistry, if you want to read more details about what I will show you tonight. Yeah. Okay. Yeah. So to start, I would like to tell you why I came to fragments. So one of my main activity for 20 years now has been to look for new biologically active compounds by virtual screening of chemical libraries. And what I observed in several campaigns is that the experimentally validated hits, they tend to be among the smallest in the selection of virtual hits. So that was kind of intriguing. And in the meantime, so for during the last 20 years, there was this new simple method approach that emerged based on the principle that fragment better fit to sub-pocket than HTS hits. So the promise is that these fragments are easily elaborated into a drug candidate. So on top of it, there was this really nice first success story. So the first drug that has been approved from fragment-based drug discovery, which is verimafanib, and it took only 6 years from the discovery of the first fragment hits to the approval by the FDA of the drug. So the first question you have to answer when you work with fragment is, what is a fragment? So if you're a practitioner in a fragment-based drug discovery, you're interested in having a compound that is soluble and stable in your biological media or buffer, and that there is a well-defined synthetic route to allow the follow-up of the discovery of the hit, from the discovery of the hit to the development of a drug candidate. And we are computing guys and would like to help the people to predict these properties. So we can find some inspiration in the literature, especially in the seminal fragment-based drug discovery publications. And you probably all know the rule of three that are the mirror of the Lipinski rule of fives. So there are other rules that have been proposed, especially there is a fluctuation over time of the size, the preferred size for fragment. So we also work on that when we start working on fragment 6 or 7 years ago. We consider that the size of the fragment may be the correct one if the fragment can retain its binding modes when it is embedded in a larger superstructural ligand. So that was the work we have done in collaboration with Lily, and here you can see Malgorzata Dval, who was postdoc who really worked a lot on this topic, and Céline Jacquemart, who was a master student at that time and who has assisted her. So Gosia, she explored the protein databank, especially the druggable pockets in the protein databank, so or in-house subset, which is called SC-PDB. And what she did is she considered the protein where we can find either a drug-like molecule or fragment. And she also looked at what we usually overlook, so the additives, which are there not for the scientific purpose, but for technical purpose. And she systematically compared the binding mode of fragment and drug-like ligand or additive and drug-like ligand. So if you're interested in looking at the data, there is a website here. I just show here the address. You can see the structure, the data, and the display of the structures. Okay, to go directly to the question of size. So there is a clear trend a clear link between the size of the fragment and the conservation of the binding mode upon elaboration into something that is bigger. It will just help you to read this picture. So on the y-axis, you have the binding mode conservation. So one is the same interactions between the fragment and the drug ligand. 0 is no common interactions. And here you have 3 panels for 3 different ranges of molecular weight. So you see clearly the trend that, okay, if you have a cutoff around 175 in Dalton, you can assume from the PDB data at least that some of the interaction will be conserved during the elaboration of the fragment. Okay, so there is an extra dimension here. So the X dimension show you the 3-dimensionality of fragments. And here it's not clear why there is a real need for 3-dimensionality to have a binding mode that is preserved. You can see here that there are some flat fragments that are whose binding mode is well conserved during the elaboration. Okay, so if we go back to our example of the first approved drug from fragment-based drug discovery, if you pay attention to the fragment, it is among the smallest one I described before. So molecular weight is around 100, and it's flat. It's a completely flat fragment. So as expected, this fragment show variable binding mode into a binding site and has multiple targets, but still it doesn't mean that it's completely useless, since it was the first information for the full program which issued the drug. So even a very small and flat fragment can bring some useful information for drug development. So you have to find the right balance. So the talk will be about library, not fragments. So the question is, what is a good library of fragment? So is this simply a collection of good fragments? Of course, it's not so simple. You see it in the title. There is no universal, ideal library. And the collection that are provided by suppliers depend on the purpose. So they are proposed for different purposes. So of course, the main purpose is discovery, but you may be interested in exploring pocket or evaluating the druggability of your protein. Then the sample you will test will also depend on the method you have chosen. So here I just suggest some of them. So typically, if you look at the first one, nuclear magnetic resonance, often use fluorine, often look signal of fluorine. So of course you will need fragments which contain fluorine. Then there are some collections that were designed for some specific targets. So there is one which is really easy to understand. It's covalent binders. So if you're interested in designing a covalent drug, then you will look for covalent fragments. Another example is here, libraries that were designed to target protein-protein interface, which are not necessarily highly druggable. Last but not least, what you expect from your library is that it will finally deliver hits. That's the most important point, actually. So here there are two points important: the complexity and the diversity in your set. So the complexity, so low complexity has been shown to be related to the versatility of your fragment. So typically, the less complex, the more chances the fragment will hit different targets. Of course, you don't want a promiscuous binder, but it's good if the fragment can target different proteins. Then about the size, what's the ideal size? I think again there is no ideal size. It depends as well on the method and the pace of the testing method. But there was a recent interesting paper who analyzed the zinc fragment set, and they could show that about 2,000 fragments will represent the full diversity of the zinc fragment database. Okay, what are the objectives of the study? First, it was, so when we started this work, we were simply interested in having an inventory of all the libraries that are available. Then we will like to check what are the fragments there, so what are their main properties. Then we analyze the chemistry within this library. So we first look at scaffolds, especially the frequency of the scaffold and whether they are simple or complex. And then we spend some time to derive a chemical map that allow to have a simple view of the chemistry that you can see with your eyes, that allows you to easily compare different libraries. Okay, so first point, what are the libraries? So we did that 1 year ago, I tell you. So that's what we could collect in 2019. So there were, so our collection is maybe not exhaustive, but okay. We try to at least cover all the supplier we work with for years. So here you see the full collection, which is composed of 81 different libraries from 14 different suppliers. So you see that some of them only provide a single fragment library, but others provide a full bench of libraries, especially enamine and live chemicals. And in total, if you merge all the fragments from all the libraries, you have about 50,000 fragments. And after removing duplicates, there is about 500,000 different fragments. So I told you before, so about so the small library, which contain about less than 2,000 fragments, may be good library for testing. So we also analyze small libraries. So if you remove all libraries which contain more than 2,000 fragments, you remove half of the libraries. So there is only 8 suppliers with 41 libraries left. And you remove a lot of compounds. Actually, if you focus on the small sized library, there are only several dozens of thousands of about 33,000 compounds. So there are many types of library, I told you. So again, we classify, so we try to find a good classification based on the annotation of the library that we can find in the catalogs or on internet. So we assign this 11 class. So I will briefly give you names according to color. So if we start on the right top corner, in yellow you have halogen enriched library. In blue we call this library general because there is no special indication about the purpose. In dark blue you have metal chelators. Here in green you have natural product-like libraries. Here in dark gray you have 3D shape libraries. In the light green you have fluorine enriched compound. The single red collection is a collection containing fragments with a high proportion of sp3 hybridized atoms. Here you have 7 collection called diverse. Here you have 1 collection that we typed PPI. Here you have 4 collections of library with covalent fragments, and all the others in magenta, purple. We assigned them the same title, the same type, because we put them together, but they were all prepared for different purposes. So here you see, if you read the annotation simply, the second one was made focus on solubility. Here you have on the bottom the smallest collection. So here you have mini fragment with a very small collection of 80 fragments. So that was for the small side libraries. So what are the types for larger library? So you see the biggest one is that one. It includes almost 200,000 compounds. So this one is very large. So you see that the representation of the types are slightly changed. So there are one type which has vanished. So dark blue was metal chelator. There is no large metal chelator libraries. There is more large sp3 rich libraries. There is only a single one diverse large library. And there are more specific libraries. For instance, here you have a collection of carboxylic acid, or a collection here of fragment with amine group. Okay. These are for the different libraries we collected. So we first plot simple properties. So you see here the sweet plots we can get out of the full collection. And okay, I think it's not a surprise if you look here, if you can draw a very nice line at 30 Dalton. Okay, there was a nice cut to generate the libraries. And I put here another threshold at 200, because if you consider this range, you have about 90% of all the fragments, all the 500,000 fragments. So on the right side, you have a second sweet plots, and this one only show you the fragment that compose the small sized library. So here you see it's only 33,000 compounds. So if you plot the same threshold, you see that, okay, the distribution of fragments is it's really shifted towards smaller fragments. So this means that the small sized library is really enriched in very small fragments. Now, if we look at the X dimension, you can see that so the fragment occupy a very narrow range of logP. So almost all the fragment end up between 0 and 2, and the distribution is quite even. So at least half of the fragments are very weakly hydrophobic. So if we look at the small size library again, we have a shift of value logP. Here the logP, they shift toward smaller value, which means that in small size library compounds tend to be more hydrophilic. So this is a general view for all fragments, but of course, if you look each library individually, you don't see the same pictures, but still you see the same trend. So here I show you again this sweet plot, and I for different library, and I put one dot for each library, considering the mean value for molecular weight and logP. And the size of the dot is related to the size of the library, the number of compounds. So again, you see that the smallest library tend to have smaller compound, but it's not always the same. So here, if you compare this one to that one, okay, you have a shift of almost 100 in molecular weight, in average molecular weight. So it's really, it's highly library dependent. So now we will move to the description of the chemical space that is covered by all these fragments. So before I go into the description, first I'll say a few words about the method. So to describe the full space, we focus on 7 types of library, representing on about 400,000 fragments. So we ignore 4 types of library, and you will understand rapidly why. So based on the fragment, we would like to derive a map that is suitable for the description of any library. So to do that, we describe the fragment with descriptor, and the descriptor we used is called ISIDA descriptor, and this has been developed in Alexander Warner's group, so who participate to that work. And so the descriptor we use is based on atoms in the fragment, and each atom is described with its surrounding, so with the different atoms which are connected following a path between 2 to 4 atoms. And then the central atom can have an extra flag depending whether it belongs to a ring or to an aromatic system. Okay, and then to have a map that is simple to interpret, we have to focus on the most frequent fragments. So we removed the fragments that are rare. So typically they are present in less than 5% of our training sets. So that's the reason why we remove some of the fragments, because there is a bias, a strong bias in the chemistry due to the presence of halogens here, or here due to the presence of warheads, which are actually not the structure which is important for the recognition of the subpocket, just for the covalent anchoring. And we also remove the PPI library because on average the size of the fragment were much higher than the size of fragment in other library. So how did we build the map? So after computing the descriptor and cleaning the set of descriptor, we project this chemical descriptor in a multidimensional space, which is called the Latin space. And we use this training set to generate what is called a manifold. So here you see on this picture, so this dimension is here in 3D, and all the descriptors are projected and they are represented by dots. And the manifold is a geometrical shape which fill the space, covering at max the points in the space. And to get the 2D map, it's an unfolding of this manifold, which gives you such map. So we also look at something that is more common, which is the scaffold, and actually you can identify into the full set about 59,000 different scaffolds using the Bemis and Murkos approach, assigning a single scaffold to each molecule. So first look at the scaffold description. So if you consider the full ensemble of scaffold, actually, yeah, you see a large majority of scaffold are present in a single fragment. And very few of them are present in very large range of fragments. Okay, so there are about 22,000 fragments that are the most interested because they cover several compounds, up to 1,000 compounds. So I just here show you the most common scaffold. For instance, benzene, it's the most common scaffold. It's found in 20,000 different fragments. It's maybe not the most interesting one. So we will ask whether is the complexity of the scaffold related to its frequency, and that's actually the case. So I just show you some statistics about the number of atoms in the scaffold. You see, the lower the number of scaffold, the most frequent the scaffold. Here, there is another description about the three-dimensionality of the scaffold. So plane of best fit is a method which fits a plane on a conformation generated for your fragment, and the score evaluate is actually the mean of the distances between the atoms, the non-hydrogen atoms, and the plane. And again, here we can see that the very frequent scaffold tend to be flatter. Last point about scaffold is the number of ring. So here you see the number of ring, 1, 2, 3, or more than 3. Again, you see a trend. So the simpler, the smaller the number of ring, the simpler the scaffold, and the more frequent it is in the library. So here, just for the example, I show you some of the most complex scaffolds. But on average, if there are several rings, they are either fused into something that is that has a compact 3D shape, or they are connected to form something that is more extended. So there are many scaffolds based on simple 5 or 6 member ring connected by simple bond or simple linkers. This example is interesting because it's the single scaffold which is actually branched with a central ring linked to more than 2 other rings. Okay, this is to give you an overview of scaffolds that are present in the set. If you want to query the scaffold, we've made a web page to give access to query. So the next and last part of my talk will be dedicated to the mapping. So the mapping here show you the projection of the different points which have been used for training the map. So Julia spent a lot of time to generate a lot of maps and to choose the best one. How did she do that? So for sure, she looked at the statistics of the learning, but she also looked at the consistency of the map. So there are there is one criteria, which is the map have to be well covered by the different compounds. So you see here it's true, there are not many holes or a few unoccupied area in the map. And you have to be able to connect the region of the map and the chemical characteristic of the compound projected into the map. So to give you an insight of this connection, here we spot different area. I will maybe just look at the first one. So this area we call R1 is made from unsaturated molecule mainly. Okay, so basically she validated that the fragment projected close to each other share some common chemical features, and this feature change if you go along a path. So they change step by step along a path. So here is another representation of the landscape where we have projected all the compounds, so the 400,000 compounds. And you see here a gradient of color which show you the likelihood of the compound, the likelihood in the different areas for one set of fragment. Okay, so this likelihood, it depicts the probability of the distribution of the data And this representation is better than the previous one, especially to compare two libraries. The reason is that the same fragment can have a non-zero probability of several points. So the projection I showed you before may be misleading. Okay, so the first question I ask here and I want to share with you is about diverse libraries. So I took two examples of diverse library, and I show you here their map. Actually, you can see that there are some common patterns between the map of the full set and the map of the two diverse library. So they all represent very populated cluster here in the same region. But still, you see there is major differences between the two diverse library. Actually, they don't cover this exactly the same space. This one on the right, although it contains less fragment, it has broad coverage of the full space. You see there is no empty space in the map, while this one is more representative of the density. Okay, so here the chemistry, which is more present on general, is also more present in this diverse set.
Esther?
So, yes?
You have 5 minutes left.
Oh, yeah, that's fine. And finish in 2 minutes. Okay, so the next library I would like to show you is the comparison between the library which focus on three-dimensionality of fragments. So here I separate all the fragments that are called 3D, and that's in the SP3 rich library. And you see again that this set do not overlap at all. So the 3-dimension set better overlap the full set, while the SP3 density mainly populates this east-north area, where here we could also find all the unsaturated cluster of molecules. Okay, the last example, we'll go briefly on these two examples. I would like simply to show you that two libraries that were built on specific features. So here you have Prestic library based on drugs, approved drugs, and here a natural product library. And again, you see that there are very specific feature of this specific library. So I put some arrows where there are some area where there is no population, red, and some area that are much more populated than in the full set. That's especially true here for natural products. You really see some rare chemistry popping up in this map. So this brings me to my conclusion. So in our study, we see that there was a wide offer of libraries, and based on the size of the library, you really have different types of compounds according to their properties. So the small size contain smaller and more hydrophobic fragments. About scaffold, we see that the simplest one were the most frequent. And I haven't shown you this, but it is in the paper. So most of the fragments contain nitrogen, oxygen is less frequent, and sulfur is even less frequent in scaffolds. And last, we have developed this method. So this maps based on the GTM methodology to evaluate different libraries, and this can allow anyone to guide either the selection of the library or the picking into a library to have its own collection. And what we will do next is to use all this method to see if the PDB information we look at for years now is really representative of what is in the library which are actually tested. So all the models and the data set are freely available. References are in the papers. So I finish here. I just want to acknowledge all the people. So again, the people involved in the work, Julia, Célian, Guillaume Brett, who helped for the online website, Su Shentang, master student, who have done some preliminary work on the project, all the team you see here, and people in Alexander Varne Club. So Gilles Marcoux worked a lot on that, and Fanny Bonachera and Dragos Sorbart, who also provide good advices, and our funders, and you for listening at such a late time today. So I'm happy to take questions if you have some.